Good morning or afternoon, everyone. This is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. Welcome to today's webinar. Thank you so much for joining us. It's presented by ASCCT and also the European Society for Toxicology in Vitro, just like this monthly series always is. Both of our organizations do a number of programs. We host meetings and pursue other activities to promote computational and in vitro toxicology. And we offer training and education and support early career scientists. You can learn more about both of our organizations on the websites on your screen. Just some general housekeeping. The webinar is recorded and the recording will be posted on the ASCCT website. And actually, let me go back there so you can see that again in a few days. Everyone will also receive a PDF copy of the presenter's slides. Your lines are all muted, and there will be an opportunity to ask questions after each presentation using the question module. So please type your question into the question box and I'll read them during the Q&A. And if you have any technical issues, you can reach us during the chat using the chat box or the question box. I just wanted to briefly give you a couple of pieces of information about some upcoming events. The Society has its annual meeting in October next month, coming up quickly. We are looking at a lot of different aspects of NAM use related to the EPA goal to move away from mammalian testing by 2035. And so, but we're also including a lot of, different folks from different areas of toxicology and different regulatory sectors that can kind of inform on that approach. Registration is open. The deadline's October 12th, and you can see the program and learn more about all of the more than 85 presentations that we'll have on all aspects of in vitro and in silico toxicology, including policy and development and implementation of NAMs. We're not going to have a webinar next month because of the meeting, but we'll have a webinar in November. We're getting that scheduled now on fish acute toxicity methods. So watch for that registration link on the website. Also, the ESTIV Society has its annual meeting in 2022. It's been postponed to 2022, but they also have a training in October. And so I'd encourage you to visit their website to find out more about that. So finally, getting to today's presentation, Dr. Johanna Nyffeler is going to be presenting phenotypic profiling for high-throughput chemical screening at the US EPA. Dr. Nyffeler is a postdoctoral grantee at the Center of Computational Toxicology and Exposure at the U.S. Environmental Protection Agency. She joined the center in September of 2017 and is focused on pipelining and analyzing data from the imaging-based high-throughput phenotypic profiling assay. She then uses these data to derive in vitro potency estimates that can be compared to in vivo data or to derive putative mode of actions. Prior to that, Dr. Nyffeler studied in Switzerland and obtained a BSc in biochemistry from the University of Freiburg and an MSc in genetics from the University of Zurich. She then pursued her PhD at the University of Konstanz, Germany. Her work was focused on developing in vitro high-content imaging assays for developmental neurotoxicology, in particular developing a migration assay for human neural crest cells. So with that, I will turn the presentation over to Dr. Nyffeler, and she can get started.
So hi everyone.
Johanna switch to the full screen mode?
Yes, I can do that. Is it better now?
Perfect, and we can hear you. So thank you. Go ahead.
Okay. Yeah, welcome everyone. Thank you for attending this webinar. I want to tell you about how we use phenotypic profiling for high throughput chemical bioactivity screening at the US EPA. And so I'm an ORISE postdoctoral grantee there. I'm going to start with saying that the views expressed in these presentations are mine and do not necessarily represent the views or policies of the agency. I also want to emphasize that I didn't do this work by myself, but this was a collaboration with a lot of people, mostly within the Center of Computational Toxicology and Exposure. And I in particular want to highlight Josh Harrell because it's his lab and he's leading this team, and he's also my mentor. And I'm highlighting on also Clinton Willis, who did a lot of the lab work, in particular the cell culture and the stainings. So the talk has several parts. First, I would like to give you a quick introduction into EPA's tier testing framework for hazard characterization. And then I will tell you what phenotypic profiling is for those of you who are not familiar with it. And then the major part of the talk, I will focus on two different applications of phenotypic profiling. In the first one, we're leveraging it to get a potency estimate for chemicals. And in the second one, we try to obtain mechanistic information. So the US EPA has released a so-called blueprint of computational toxicology, in which they propose a tier testing framework for hazard characterization of chemicals. And I'll just get my pointer. And so it has several tiers, and the idea is that we have a lot of chemicals that we want to test, and we start with having some chemical structure information, and we would run some broad coverage, high content assays. And depending on the result, if we do see activity in no defined target, biological target, we can go this route, or if we see, if we get indication that particular targets are hit, we would follow up with selected in vitro assays, like lower tier assays. And so our lab is based on top here, what is shown in the red circle. And we have two profiling assays that we're currently running. The first one is transcriptomics, and the second one is phenotypic profiling. And this will be the one that I focus on for the remainder of this talk. I struggle to advance my slides. So what is phenotypic profiling, for those of you who are not familiar with it? So profiling assays are different from more well-known targeted assays. So in targeted assays, you measure usually one or a few endpoints. I picked here as an example an estrogen receptor agonist assay of the ToxCast battery. And it works by measuring radioligand binding. So if a chemical is an agonist, it will lead to a decrease in radioligand binding. And then you have a positive control, in this case 17-beta estradiol. So in this type of assay, you know the response is a predictable change in a single endpoint in a known direction. And this is in contrast to profiling assays. Here is an example, transcriptomics, where you basically have thousands to 10,000s of kind of endpoints, and the response can be any meaningful change in transcript levels. So for active chemicals, the response can involve changes in many different endpoints in unknown directions, and it can also be different from chemical to chemical. So you can get very different information out of that, but it's also making data analysis quite challenging. Now, imaging-based phenotypic profiling is a profiling method where you stain various cell organelles with fluorescent dyes in vitro cultures. And then we can assess a large variety of morphological features in these individual cells. So we are using a version or a combination of labels that is called the cell painting assay that was developed by the Broad Institute. And we stain the nucleus, the nucleoli, the endoplasmic reticulum, the Golgi, the membrane, and the actin skeleton, and the mitochondria. And then we image the plates, and we derive a lot of different features, such as the shape of the organelles, the intensity of the label, the localization, or the texture. In our version of the assay, we derive 1,300 features, and we call our version of the assay high-throughput phenotypic profiling, or HTPP. Here are some example images. On the left side, you can see in panel A, untreated cells labeled for the nucleus in blue and for the mitochondria in red and whitish. And so you can see that the mitochondria sit next to the nuclei, or to the nucleus. And then in panel B, you can see cells that are treated with berberine chloride. And you can see that the mitochondrial staining is different. So it's displayed here in white, which means just a high intensity, and it's more granular. So we refer to that as a change in mitochondrial compactness or texture. And on the right side is a different example where we labeled or we display different channels. And you can see in panel E, untreated cells, and in panel F, cells treated with etoposide. And you can see that these cells are much larger. So the cells, they were swelling. And so these are just two examples of strong phenotypes that we can observe qualitatively. But with our data analysis, we can also detect much smaller changes that wouldn't be easily observable by eye. So the way we run this assay is that we plate the cells and then we let them attach for 24 hours. We dispense the chemicals and expose the cells for another 24 hours. And then we live label the cells, fix them, and permeabilize and label them. So for our first study, we used a parallel set of plates. And the first set of, so where we treated, that were treated with the same chemicals. And so the first set of plate was used to measure cell viability by labeling with Hoechst and propediomyodide. So propediomyodide marks the dying cells. So you see an increase in the signal if cells are dying. And then in our actual cell painting plates, we label them with this cocktail of reagents and then image the cells. We segment the cells, profile the different compartments, and we get cell level data for each of the 1,300 features. So then we do some data reduction and normalization to end up with well level data. So to get one value for each well for each feature. And then we can perform dose response modeling. There are very different ways how to do that, but in the end we want to come up with a potency estimate. So, and that's what we refer to as an in vitro point of departure, POD, or a phenotype altering concentration, PAC. So we've successfully run this assay, and here I show you that the behavior of reference chemicals. So on every individual plate that we screen, we run this set of reference chemicals in concentration response. Here I just show you one concentration for each chemical, but this is so the columns are the 1,300 different features, and the rows are different replications of the same chemical. And what you can see is that we get very reproducible profiles among the different replicate, but also that the profiles of different chemicals look different from each other. And this is also highlighted here in panel C, This is a correlation matrix, and we can see that the different replicates of the same chemical look very similar to each other, but different from other chemicals. And in panel D, we can see how reproducible the potency estimates are that we obtain. They are usually less, the variation is usually less than an order of magnitude, which is pretty good. So having set up the assay, we can now use it for our applications. So in this first application, I want to show you how we use it for potency estimation. And this work has been published earlier this year in the journal Toxicology and Applied Pharmacology. For this study, we screened a set of 460 environmental chemicals. Most of them are pesticides, but there were some drug-like chemicals, food additives, and industrial chemicals. Most of these chemicals were also from the so-called UPCRA list. And so the UPCRA list is a set of chemicals that has available in vivo effect values and also the necessary toxicokinetic parameters so that we can do in vitro to in vivo extrapolation. And this will be useful later when we want to compare our in vitro results to existing in vivo data. So for this experiment, we've used U2 as osteosarcoma cells. We've exposed the cells for 24 hours. We tested 8 concentrations per chemical, and we've done 4 independent experiments. But when we screened all these chemicals, we had to think, how do we analyze this data? So we obtained 1,300 features from each well, and there are different ways how you can derive a potency estimate. And this hasn't been really done before for cell painting. It's been done for transcriptomics, so we try to leverage that information a bit, but there are also some important differences from cell painting to transcriptomics. So we ended up doing a comparison of 15 different analysis approaches to see which one would give us the best results. I'm not going into that in detail. This work has also been published earlier this year. I just want to show that there are different ways how you can derive a potency estimate. For example, you can fit one concentration response curve to each feature and then somehow aggregate that and define a potency. Or you can do some aggregation first of the data and then fit a fewer number of curves. For example, here, we aggregated the data first within category feature categories and then fitted some curves. Or you can do a more drastic approach and combine all the 1,300 features into one number and then fit only one curve. So there are different ways how you can do that. The take-home message here is that we've tested all these different approaches, and it turns out that no matter which approach we use, there were always 50 to 70% of the chemicals were identified as active. And it was also like a large proportion of the chemicals were identified as inactive by all approaches, and another set was identified as active with all the approaches. But we ended up deciding for this project to go forward with 2 approaches. So in the first approach, we always start with the 1,300 features. But in the first approach, you boil them down to one number, and that is a Mahalanobis distance. So a Mahalanobis distance is similar to a Euclidean distance, which is really just the distance in space. So if you imagine the 1,300 features in a 1,300 dimensional space, and then we really just measure the distance of the well of interest with control wells. So a larger distance means that the phenotype of this chemical is more far away from control cells. So we just fit one curve for each chemical that gives us one benchmark concentration. And then we say that this is the phenotype aldrin concentration. In the second approach, we group these 1,300 features into 49 categories. So each feature belongs to exactly one feature category. And then we do the same procedure with the Mahalanobis distance, but within categories. So that results in 49 curves that are fit, so potentially 49 benchmark concentrations, and we picked the most sensitive one as the phenotype altering concentration. And then we decided to call a chemical active if it was active with either one of these two methods. And if we use this strategy, 70% of the chemicals were active, as indicated here on the right. For the chemicals that were active, we then went on and compared the potency estimates to in vivo data and exposure. So here, it's a busy slide, but I will walk you through it. So we start here with the HTPP point of departure, which is in micromolar. But if we want to compare it to in vivo data is typically in milligrams per kilogram body weight per day. To compare that, we have to convert our micromolar concentration using in vitro to in vivo extrapolation. And we did that with the high-throughput toxicokinetics package, HTTK. So what we want to do is we want to estimate which dose a human would need to take to end up with a plasma concentration that corresponds to this point of departure. And we model this, we get a distribution because we take into account that the inter-individual differences exist in pharmacokinetics. So we get here a distribution. But then we can, and this dose, we call it administered equivalent dose. And we can then compare this dose to in vivo effect values. So here we chose in vivo effect values that were in the database called ToxValDB. And we summarized all the values for a single chemical by just choosing the 5th percentile of all the effect values to try to pick a conservative estimate. And on the other hand, we can compare our administered equivalent dose to exposure estimates. So we have exposure predictions for these chemicals from the EPA XPOCast project. And there we would compare them to a higher level of exposure, again, to be conservative. And here in between, we can also see that there are other new approach methodologies, and we can compare our results to those ones too. So this is the result of these 300 chemicals, when for each chemical we compare the potency estimate from the NAMs to the potency estimate from in vivo effect values. And so the black line indicates the unity line. And to the left means that NAMs were more conservative, and to the right means that the NAM was less conservative. So the first thing that you can see is that the purple histogram, which is the HTPP assay, is quite symmetric around this unity line. And overall, for 80% of the chemicals, the HTPP dose is within 2 orders of magnitude of the in vivo point of departure. And I think that is actually quite amazing that we have a simple cellular system using just one cell type, and we apply these different labels, and we do a pretty good job at predicting which toxicity or which effects we could see in vivo. Not effects, but like which potency we could observe in vivo. You can also see that the blue histogram, which is the ToxCast data, is shifted to the left. So it generally has lower values. And the threshold of toxicological concern in green is even more shifted to the left. And that is well known that this is gives generally protective estimates, but they are they're not very predictive of the in vivo toxicity. This is a different view of the same data, where I've also overlaid our variability in our estimation. And then I wanted to see whether where the traditional point of departure falls in relation to this estimated range. And what we can see is that for a quarter of the chemicals, the HTPP estimate is above the traditional point of departure. And that's not really good. But for 3/4 of the cases, we are either giving a comparable surrogate or a conservative surrogate for the in vivo value. And ToxCast has even more cases, and the TDC even more. That's what we've seen on the slide before. But so overall, for 72% of chemicals, we would have obtained a conservative or comparable surrogate. So we can also compare our bioactivity thresholds with exposure estimates. And to do that, we calculate a so-called bioactivity exposure ratio, or BER, by simply dividing the lower bound of the HTPP bioactivity with the upper bound of the exposure estimate. And so this is indicated here in the histogram, again, showing the 300 chemicals. And this is the unity line. So chemicals to the left are chemicals for which we see bioactivity only at concentrations that are higher than estimated exposure. And you can see that for most of the chemicals, they are shifted to the right, and on average, they are 3 orders of magnitude above the exposure estimate. So we usually only see bioactivity at concentrations that are 1,000 times higher than what humans are exposed to. But there is also a small subset of chemical here to the left for which we do see bioactivity at levels that humans could be exposed to. And that is indicated on the right. So that's the same data, just represented in a different way, where you can see that there's a subset of chemicals that are above the solid line here. And we think that for these chemicals, there is the potential for humans to be exposed to bioactive concentrations. So this concludes the first application. So I think I've shown that HTPP in vitro potencies can be used for prioritization of chemicals based on inferred bioactivity in relation to predicted human exposure. The next step will now be to test these chemicals in multiple cell types to increase biological coverage. So the idea is that for those chemicals where we did a poor job in predicting the in vivo toxicity, that if we would test more different cell types, we might be able to pick up more biology and we would get a better estimate. So now I'd like to move to the second application, which is the identification of putative mode of actions. And this is very much work in progress. Towards the end, it's not very developed yet, I would want to say. So for this project, we did a second screen. This time we screened 1,200 chemicals. Many of these chemicals are of interest to the agency under the TSCA requirement. Also, most of the chemicals in the first screen were screened again here. But more interestingly, 180 chemicals were annotated with a particular target in our database called RefChemDB. So we've done this experiment again in U2S cells. We've exposed them for 24 hours. We used again 8 concentrations and have done 4 independent experiments. Before I'm going or showing you the mechanistic interpretation, I thought I'd do a quick comparison to the ToxCast assay results, because nearly all of these chemicals have also been tested in the ToxCast screening battery. What you can see here on the left is that in HTPP, a bit less than half of the chemicals were active. And on the right, you can see that the chemicals that were active in HTPP, they tend to have also a high frequency of being active in ToxCast assays. So we call them as having a high hit rate. While chemicals that are inactive in HTPP, they generally have a lower hit rate in ToxCast, and many of them have very few hits in these assays. So overall, I would say that chemicals that are active in HTPP are more often promiscuous in ToxCast. I then wanted to compare the potencies, and we've also seen it a little bit in the previous project, that we know that we are getting less potent values than ToxCast predictions. And that is shown here again, where I simply compare the HTPP phenotype altering concentration to a point of departure that I derived from the ToxCast assays. And what you can see is that we are getting less potent values than the ToxCast point of departures. I've also compared them to the ToxCast cytotoxicity burst estimate. So there is a subset of assays in the ToxCast battery that are more associated with cytotoxicity or cell viability, and you can get a potency estimate using only those assays. And in that case, HTPP gives us more potent estimates than just the cytotoxicity burst estimate. So that means we're not doing as good as ToxCast, kind of, but we're still doing better than if you would use general cytotoxicity assays. But yeah, here I also want to show that how reproducible the assay is. So for this screen, we decided to use a different set of reference chemicals. We chose dexamethasone, a glucocorticoid, and retinoic acid, a retinoid, to be included as reference chemicals on every single plate, again in concentration response. And here I show you the profiles of these chemicals across 29 plate groups. And you can see that we get very reproducible profiles. And again, the profiles of the chemicals look different from each other. So I want to show you here, as an example, the results of a subset of chemicals, and these are all nuclear receptor modulators. So I've mentioned before that around 180 chemicals were annotated with a target, and so of those, 52 chemicals were annotated as targeting a nuclear receptor. So they can either be agonists or inhibitors, or sometimes we didn't know the direction. And so the nuclear receptors that were targets are the ones outlined here on the left. And when I've done the scatter plot that I've showed before for comparing the HTPP pack to the ToxCast potency, but just for these 52 chemicals, you can see that there are two groups of chemicals. So there's a big group here for which we really do a poor prediction. But there's a small subset circled here for which actually the HTPP potency line up quite well with the ToxCast potencies. And it also turns out that these chemicals belong to two particular targets. They belong either to the glucocorticoids or retinoic acid receptor modulators. So we thought maybe this has something to do with the genes that are expressed, because if a target is not expressed, obviously the chemical cannot act via that receptor. So we looked at some gene expression data for the U2S cells, and we could confirm that. So the two receptors for which we did good predictions, they were expressed in U2S cells. But for example, estrogen receptors are not expressed in U2S cells. And so we did a really bad job at predicting those ones. And that's not surprising if the receptor is not present. Next, we finally wanted to look at the profiles and compare the profiles of different chemicals. So here to the left, you can see a correlation matrix where I compared the phenotypic profiles of each of the chemicals against all the other chemicals. And they are ordered here by the color code according to their target. And so what you can see is that there are two groups of chemicals for which the different modulators gave very similar phenotypic profiles. And these are, again, the glucocorticoid modulators and the retinoids, as well as the vitamin D agonists. So they give very characteristic profiles that look different from the other chemicals. And again, that lines up well with gene expression. But the fact that a gene is expressed does not guarantee that we see a characteristic profile. For example, we have the PPARs here. They are obviously expressed in U2S cells, and yet we did not get any characteristic profiles for these modulators. So this was just, so my conclusion from that is that if we want to make predictions about mechanisms, we should only make predictions if we see multiple reference chemicals, or like, annotated chemicals having the same characteristic profiles. Because if they all have different profiles, that indicates to me that they are more likely hitting an off-target effect. So we definitely need to make sure that we get multiple chemicals with the same profile, so that we would have any confidence that we can find about our predictions. So this is exactly what we wanted to use for this next part. So having identified several chemicals that act, or we know that we have several retinoids in the system, and we've identified that they all have very similar biological profiles, we wanted to see if we find test chemicals that were not annotated with targeting the retinoic acid receptors that have a similar profile. And we were able to identify 5 test chemicals. And I also thought maybe they just look similar because they are bioactive. Maybe they are just looking similar to a lot of different things. So I plotted here for each chemical the similarity to the 100 nearest chemicals. And you see in red their similarity to the known retinoids, and in orange to the other, what we call candidate retinoids. And you can see for these 5 chemicals that they look similar to these known retinoids and candidate retinoids, but they do not look similar to other chemicals. So that is a specific profile. I also convinced myself by qualitatively looking at the profiles. So you have here again in columns the 1,300 different features. And here in bold are the different concentrations, the active concentrations of the known retinoids. And here below you see the active concentrations of the suspected retinoids. And to me, this shows that there is a characteristic profile and that these chemicals indeed do look similar, but they have a bit of weaker profile than the known retinoids. After that, I was really curious if the activity of so I really hypothesized that these chemicals affect the retinoic acid pathway. And I was curious whether this was already detected in the ToxCast assay battery. So I pulled up the data for these chemicals. What you can see here on the right is the number of assays that the chemical was tested in, and the number of assays where the chemical was found to be active. And for each active assay, you see a point to the left with the potency. And I've highlighted specific assays with colors. So the assays that target the retinoic acid receptors and the RXR pathway are highlighted, as well as in red is an assay that is based on a stem cell model that is sensitive to retinoic acid pathway disturbance. And in purple, I highlighted the potency that we found in HTPP. And you can see that the known retinoids, they are active in one or another of these targeted ToxCast assays. And as I've seen before, that our HTPP potency lines up quite well with that. But for our suspected retinoids, they had barely any activity in these assays. So there is triphenyl tin hydroxide, which was active. And it actually turns out, upon looking at the literature, that it's a known RXR agonist. So that was also reflected by the results in ToxCast. So that was also properly identified with our HTPP. So that's reassuring. There's one other chemical that had some activity in the stem cell assay, but for the other 3 chemicals, there was no activity in the respective ToxCast assays. So to me, that means that HTPP could yield complementary information to existing assays, and that's quite exciting to me. Of course, on the long run, at EPA, we are more interested into effects of non-drug-like chemicals. So the work that I've shown up to now is more proof of concept. So pharmaceutical companies have applied cell painting assay previously on drug-like chemicals, and that works quite well. But we are more interested into non-drug-like chemicals. And this is very much work in progress. So here I just have some qualitative results. For example, on top here, we have a group of organochlorines, and I've indicated their profiles. And you can see that they all affect the DNA compartment, the nuclear compartment, in a similar manner. And on the bottom, we see a group of strobilurins, and they have effects in various channels, but most pronounced in the mitochondrial channel. So we do have indication that certain groups of environmental chemicals display characteristic profiles. So to conclude the second part, chemicals with different mode of action display characteristic profiles. We've seen that, for example, for the glucocorticoids and the retinoids. We can identify test chemicals that are biologically similar to annotated chemicals. We've shown that with the retinoids. And we've also seen that certain groups of environmental chemicals display characteristic profiles. So overall, we can conclude that HTPP can potentially be used to derive mode of action information and help in prioritization of lower tier follow-up assays. The next steps are to confirm the suspected retinoids in an orthogonal assay. So we decided to choose qPCR, and this work is at the moment ongoing. And we also want to look at how well does structural similarity translate into biological similarity. With that, I'm at the end of my presentation, and I thank you for your attention. I'm happy to take questions, and also feel free to reach out by email if there's anything else.
Thank you very much, Dr. Nyffeler. We have a few questions coming in already. So just to remind everyone, if you have a question, you can put it in the question box on the GoToWebinar control panel on your screen, and we will ask it. So the first question came in a little bit early, and so I think the answer might be yes, but I want to see, you know, what if you have more comments about this. So they're saying that using HTTP data to prioritize, you know, large inventories of chemicals seems brilliant, actually. How about application of this idea into risk assessment of individual chemicals? So maybe digging into the toxicity of groups of chemicals or individual chemicals.
Yeah, I'm not sure how much it could be directly applied for risk assessment, because from what I understand is that they are interested in adverse effects, and with the cell painting assay at this point, we don't know which effects are adverse versus adaptive. So we can just see changes. We can see that the cells are affected, but we don't know whether that is bad. And from my understanding, the risk assessors want to know that something is bad so that they could use it. But the idea is our assay can be used for prioritization and can inform the lower tier assays so that we can then get more information that will then be useful for risk assessment.
So it's sort of a weight of evidence approach, which is kind of what you presented in the beginning there. Lots of information.
Yeah, I think you would definitely need other assays. I don't think it's likely that they would say, oh, we see that the cell morphology is changed in one cell type, let's regulate on that. I would be happy if they would do so, but I don't think it's realistic.
A question about how you analyze your data. Do you use specific kinds of software that you found really helpful?
Yeah, so we extract the features using the software that is from the same company that we have the microscope of. So that's the Harmony software. But there are also open source software available to do the feature extraction, and the most well-known one is Cell Profiler. And there are actually a lot of software now around that would do feature extraction for you. And then once you have the feature, you have to deal with all the data. So that is all R code that I wrote. And then we have concentration response modeling, and there are again several ways how you can do concentration response modeling. And we used either the BMD Express software that is open source, or a new version of the TCPL package from EPA that is also open source. Okay, and then yeah, to in vivo extrapolation is also an R package. So yeah, I think the data analysis, the data wrangling is quite extensive for these profiling assays. So running the assay is not very hard, but then you have to deal with all these gigabytes of data, and that's not very easy.
What about chemicals requiring metabolism, either I think to be active or some might be inactivated?
Yeah, so that's a good point. So the current way how we run the assay is just a simple in vitro assay and will not account for metabolism. But there is another group at EPA working on assays to incorporate metabolism. And the idea is that we can then couple these two assays so that we could couple metabolism and cell painting assay, and then we could account for that, but not at the present version.
Okay. Are there plans to extend the analysis to more cell models or different exposure protocols?
Yes, absolutely. So we prioritize different cell models over different exposure periods because we've done a pilot experiment and we found that the point of departures are relatively constant across time, at least for this small set of chemicals. But definitely we think testing different cell types will be beneficial. So we are working on finding a good panel of different cell types that would give us the most diverse phenotypes, basically, or cells that look already different and that also express a different set of genes, so that hopefully the cells would react to different perturbations. So yes, this is definitely going on, and in the near future we will be screening in different cell types.
Great.
And we've already done the preliminary work, like we know we can run the assay in different cell types. We just haven't run the hundreds of chemicals.
Okay. Someone's wondering if you looked more closely at the 28% of chemicals where the in vitro HTTP potency was greater than the in vivo point of departure. And was
yes.
it unique about these chemicals or the?
Yes, we did that.
Sorry.
And that's also described in the manuscript. So we've looked if certain chemical features are enriched among these chemicals for which we did a poor prediction. And what we found was that we did a really poor job in predicting organophosphates, and I believe an other group of chemicals that I forgot. So yes, we were able to see that there is a group of chemicals that when we see the structure, we can already say we will not do a good prediction with those. And it's not surprising because these organophosphates, they act on a receptor that is not expressed in U2S cells. So it's not surprising that we do a bad job at predicting them. And that's exactly also why it would be good to have different cell models. So if we have a cell model that expresses this receptor, we would hope to pick up this effect.
Great. A quick question, how many chemicals can you or do you typically test simultaneously?
So we've done several hundred at the same time in a screening session. Also the 1200 chemicals we've tested them in 2 batches actually. So yeah, we can probably do 600-700 at the same time. And that takes us a couple of weeks. So actually for one screening block, we take maybe 3 weeks of wet lab work and then maybe another 2 weeks to image all the plates, and then a couple of weeks for data analysis.
And is that a manual process or do you have some automation?
Yeah, so we do have quite a lot of robotics that help us to see the cells, to label the cells, but at this point, we still have to handle the plates by ourselves. Like, I mean, we have to place them in the machine and start the machine and things like this. We also have to put every plate in the microscope. We are working on getting this more automated so that there will be less manual work involved.
Mm-hmm.
So there's not really pipetting that we need to do, but it's like walking plates around from one place to the other and starting the machine.
A question about labeling. How did you manage to separate organelle features labeled with the same dye?
Yes, so we have to admit we cannot perfectly separate them. They are in the same channel and they have the same excitation and extinction, so we will not be able to properly separate them. So they are mostly separated by the different localization within the cell. So in the green channel, we have the nucleoli and we have the endoplasmic reticulum. And for that one, we simply say that the green signal that we see in the nucleus, we attribute that to the nucleoli, and the green signal in the cytoplasm, we attribute it to being a part of the endoplasmic reticulum stain. But yeah, for the orange channel, we have the Golgi and the cytoskeleton. They're both in the cytoplasm. So we cannot really distinguish the different organelles. Yeah, that's also why I refer often to more as channels instead of organelles. It's more when you look at the pictures and then you can say, okay, this seems more to be a problem of the Golgi, or this is more a problem of the cytoskeleton. But this is not perfectly separable.
Okay. There's a question about mixtures. Someone's working with mixtures and they're wondering if you've had any experience using mixtures, or are you mostly using single chemicals?
We haven't done any mixtures, but I would assume that this technique would work well for mixtures, because you can just, well, you can, on one hand, you can just see at which concentration you see bioactivity without needing to know any particular mode of action. Or yeah, you could then later look at the profiles and see, maybe you could even say which one of the chemicals in the mixture is producing the effect, maybe, but we haven't tried that.
And just a comment coming in on the last question that low band pass filters could help separate signal for fluorophores with similar emission or excitation wavelengths.
Yeah, thank you. Yeah, it's a problem when you have 7, I think we have 7 organelles and 6 fluorophores, something like that, and only 4.5 channels actually to image. So
Okay, well, we have thrown a ton of questions at you, so thank you so much for sharing your expertise. There is obviously a lot of interest in your work and the methods that you're developing. So thank you again, and I think we'll end the webinar. Thanks everyone for listening. Thanks for your presentation, Dr. Nyffeler.
And thank you for inviting me.
Have a great rest of your day.
Bye, have a good day.
Bye, thanks.